Hello my dear students, how are you all? Today we are going to learn very important topic, that is role of enzymes in pathogenesis. Why I am saying this is an important topic means this topic is placed in ARS syllabus, that is under the chapter 3 molecular host physiology and molecular plant pathology, and who are preparing for agricultural research service in plant pathology. For them this topic is very important because you may get one or two questions from this topic. The role of toxins in relation to plant disease already I covered in my previous video. So today we are going to learn about the role of enzymes in the plant disease. So before understanding the different enzymes produced by the pathogens, first we have to understand the basic structure of the cell wall. So first of all, what is enzyme? Enzymes are nothing but large protein molecules which can catalyze all interrelated reactions in a living cell. So most pathogens, they derive their energy principally from this enzymatic breakdown of the food materials whatever present in the host tissue. So first we will understand what is the composition of cell wall and what are the different parts associated with this cell wall. Cell wall is functionally divided into three regions. Already we know about it, that is middle lamella. That middle lamella is majorly made up of the pectin substances, and primary cell wall, that is which is made up of cellulose and pectic substances, and secondary cell wall, that is entirely cellulose. So the middle lamella act as a intercellular cement which binds all the cells together in a tissue system. So middle lamella is majorly responsible for binding of all the cells together, and pectin or pectic substances which are present in the middle lamella, these are chemical constituents of wall layer, and this entire middle lamella, whereas in other layers like cellulose is found in good amounts, like primary cell wall and secondary cell wall we can find the cellulose, but middle lamella is majorly made up of the pectin or pectic substances. Here you can see the cell wall structure. So what are the other components of the cell wall are hemicellulose, lignin and some amount of proteins. I will explain each one in coming slides. So main components of cell wall are pectic substances, cellulose, hemicelluloses, lignin and small quantity of proteins. So how pathogen produce the different enzymes to degrade all these substances to make its an entry into the host cell, because if it has to make entry means it has to degrade all these components, that means it should provide some enzymes to degrade these components. If you will see the picture for clear understanding of viewer, I gave picture here. You can see the cell wall structure. This is just cross section here I shown here. Then first one is the cuticle, and after before cuticle wax layer used to be there. That wax layer used to be embedded in the cuticle. Then wax lamella and cutin, cellulose lamella and pectin lamella. That pectin used to be present in the middle lamella. Then cellulose layer, that is secondary cell wall and plasma membrane, and inside it is the cytoplasm. I think you can understand the cell wall structure basically, so that you will get overall idea how the pathogen it can make its move to degrade these substances one by one. First one is the epidermis of plant. That epidermis is covered by the cuticle. All we know that plant even that cell wall is outermost layer used to be the cuticle, whose major chemical substances is cutin in addition to cuticular wax. That cuticular wax already I told that it used to be embedded in the cutin. So first we will see what is the cuticular wax. These plant waxes generally they are found as a granular or rod like projections or as a continuous layer outside or within the cuticle. So in this cuticle only this cuticular wax used to be present like rod like projections, and wax formation is a continuous process and it is not a terminal phase in the development of leaf. It So during the maturation and development phase, this cuticular wax formation, it used to be continuously formed. It is not like that at one stage it forms and later it stopped its formation. It is a continuous process. And so this cuticular wax, what it is constituents, that cuticular wax are made up of these long chain molecules of paraffin, hydrocarbons and alcohols, ketones and acids. So cuticular wax is made up of the paraffin, hydrocarbons, alcohols, ketones and acids. And most of the fungi and parasitic higher plants, then how they will penetrate these wax layers means because of mechanical force only. Suppose during the development of the germ tube through aprisoria formation, I think I already explained in some previous sessions. So during that force they will apply some mechanical force to break down this cuticular wax. So here there is no need for the production of any enzymes. Only through by applying the mechanical force the pathogen make its an entry through this cuticular wax. Coming to the cutin, what is cutin? Cutin is insoluble polyester of unbranched derivatives of either C16 or C18 hydroxy fatty acids. There are saturated and unsaturated fatty acids, but here I am not going into the very detailed explanation of what is the C16, C18 hydroxy fatty acids. I think you know about this palmitic acid, these fatty acids. I will make another video about these fatty acids. Okay, then when cutin is admixed with waxes on the upper side and with pectin and cellulose on the lower side. So before upper side of that cutin, what it is used to be there, that waxes, cuticular waxes, on lower side pectin and cellulose. Then cutinases break the cutin molecules. So here pathogen has to produce the cutinases. These are the enzymes which can capable of breaking the cutin molecules. And then after breaking of these cutin molecules, these cutinases release the monomers as well as oligomers from insoluble cutin polymer. These monomers and oligomers, these are the polysaccharides which are in the continuous chain formation. Then cutinases, it break cutin molecules because cutin is a continuous chain of polysaccharide. It will release these monomers and oligomers. Then where this cutinase reaches at highest concentration means at pertaining the point of germ tube and at the infection peg of aprisorium forming fungi. So when the pathogen during the germination, that spore germination, when it is producing aprisorium and penetrating peg, if you will remember that structure, aprisorium formation, then it will produce penetration peg. During that time what it will happen, plant receives the signals from the pathogen, then it will activate its system that some foreign particle is trying to make entry into the cell wall, so it has to resist itself. Then it will increase the cutinase cutin production also. So then cutinases means this pathogen will produce more cutinases because it has to make an entry, because cutin is a strong substance. So cutinases will be more and automatically it will break the cutin, then it makes entry into the cell wall. And where we can see this type of entry means Coletrotrichum gliosporides, which used to cause anthracnose disease, and Spirotheca panosa and Vencheria inaqualis, that apple scab disease, and Helminthosporium victoria. In maize we can see the production of these cutinases. And coming to the second one is the pectic substances, where we can find these pectic substances in the middle lamella. That middle lamella is rich in pectic substances. So these are the major components of the middle lamella. That is, middle lamella is connecting all the cells together in a cell tissue. So they also make up of large portion of primary cell wall, in which they form an amorphous gel filling the spaces between cellulose microfibrils. So not only in middle lamella, these pectic substances also present in the primary cell wall, and also these pectic substances fill the space between the microfibrils. This cellulose generally used to be present in the form of microfibrils. This microfibrils picture I will show in the next slides for your understanding purpose. But here just you remember that pectin substances Beside middle lamella, it is also present in the primary cell wall, and also it fill the spaces between the cellulose microfibrils. And coming to the this chemical composition, what it is exactly pectin? Pectin is a polysaccharide. These polysaccharides are the compounds which consisting mostly of the D-galacturonic acid units with alpha 1,4 glycosidic bonds. So the pectin substances are nothing but polysaccharides. They are having the D-galacturonic acid and alpha 1,4 glycosidic bonds. And these pectic substances, what they will do, they will esterified with methyl groups. So that means they will form a chemical reaction with the methyl groups, or they linked with other carboxyl groups in calcium and magnesium salt bridges. And these pectin substances are, there are three types based on this esterification with the methyl groups or other chemical carboxyl groups. So what are those three types of pectic substances are? First one is the pectic acid, which is a non-methylated units, and another one, second one is the pectinic acid, where it is less than 75% methylated galacturon units used to be there, and third one is the pectin, where more than 75% methylated units used to be there. So another one term is there, that is protopectin. So what when we will use this protopectin term, this is used to denote substances which are soluble in water, and upon restricted hydrolysis they will yield the pectinic acid. So this protopectin will undergo hydrolysis and it will release the pectinic acid. And the enzymes which can degrade these pectic substances, what we used to call automatically, it is a pectinases or pectolytic enzymes. So we can call them as a pectinases or pectolytic enzymes. So there are different pectinases or pectolytic enzymes which are produced by the fungi. Let us see what are those pectinases. First one is the pectin methyl esterase, and second one is the polygalacturonases, that is PGs, and third one is the pectin lyases, that is PLs. So what is the role of each enzyme during the pathogenesis? This pectin methyl esterases, the name itself indicating that it will remove the methyl groups from the pectin substances. So pectin methyl esterases breaks the ester bonds and removes the methyl groups from the pectin, which leading to the formation of pectic acid and methanol. Methanol is CH3OH, and whereas ethanol C2H5OH. So here it will release the methanol separately, that is the pectin methyl esterases it will do. And second one is the polygalacturonases. What polygalacturonases will do? It will split the pectin chain by adding a molecule of water, and it will break the linkage between two galacturon units, and they catalyze reactions that break the alpha 1,4 glycosidic bonds, because pectin is made up of alpha 1,4 glycosidic bonds. So polygalacturonases, they will add the molecule of water and they will catalyze the reactions which can break the alpha 1,4 glycosidic bonds. Coming to the third one, pectin lyases, how they will split the pectin means they will split the pectin chain by removing the molecule of water from the linkage and thereby breaking it and releasing the products with unsaturated double bonds. Whereas polygalacturonases split the chain by adding a molecule of water, whereas pectin lyases split the pectin chain by removing a molecule of water from the linkages. And these pectin enzymes can be, that is again two types, exopeptidases which can break only the terminal linkages in the pectins, or endopectinases which can break the pectin chain at random sites, that is internal, not at the terminal sites. Internally it can break at a random site, means at any place it can break. These are called endopectinases. So what will happen after pectin degradation? Simple, it is results in the liquefaction of the pectin substances, and it will weak the cell walls, because pectin is the strong substance present in the middle lamella. If pectin is degraded, automatically it will lead to the tissue maceration. If in our brain, if we are thinking about tissue maceration, this tissue maceration will be caused by only the soft rot fungi. and soft rot bacteria. Soft rot bacteria means, example, we have to remember about Erwinia Keratovera subspecies Keratovera. We have to be get on the tips about these examples. Soft rot bacteria, that means Erwinia Keratovera subspecies Keratovera, and fungi like Botrytis cinerea and Sclerotium ralsei. These are the fungi and bacteria which produce the pectinases. So after pectin, then coming to the cellulose. Up to now we learned about the cuticular wax, cuticle and pectin. Now coming to the cellulose. First we will learn about what it is cellulose and what are the different components making the cellulose and what are the different enzymes required to break the cellulose. So cellulose is it is also a polysaccharide. All these compounds are nothing but polysaccharides, and this cellulose is made of chains of beta D glucopyranose units, and that is 1,4 linkage of beta glucose monomers. Then glucose chains, these are held by hydrogen bonds. As we know that in cellulose these glucose chains are generally they are held together by hydrogen bonds. And cellulose we can see in all the higher plants as a skeletal substance of cell wall in the form of microfibrils. That means cellulose used to be present in the form of microfibrils, and primary and secondary wall also consist of the matrix. In that matrix these microfibrils used to be embedded, and these microfibrils used to be present like a bundles of iron bars which are in a reinforced concrete building. That means they are arranged one by one, that is in a continuous chain, or they may arranged in non-order fashion. If they are arranged in an orderly fashion, then it will give a crystalline form. If in not in orderly fashion, then it will give a amorphous form. But crystalline form is more stronger than the amorphous form. Here you can see that microfibrils, that cellulose and microfibrils, how they are arranged in order fashion. If they are arranged like in this order fashion, then they are in crystalline form, which will give more resistant of the host to the pathogen. But if it is in less order form than amorphous form, then it can not offer that much resistant to the pathogen, so that pathogen can enter very easily. I think now you understood about the microfibrils. And here in this picture also you can clearly understand that here you see this is the middle lamella where we can see the pectin, and these are the cellulose microfibrils. And here we can observe some other structures like hemicellulose. Here you can see hemicellulose is present between these cellulose microfibrils, and some soluble proteins are also attached to these microfibrils and they are floating inside. So beside these cellulose microfibrils we can see the hemicellulose and soluble proteins. So we will study about these hemicellulose and soluble proteins also. That is, a plant cell wall is arranged in layers and contains cellulose microfibrils, hemicellulose, pectin, lignin and soluble proteins. So the space between microfibrils and between these cellulose chains, that is filled with pectins, hemicellulose and also lignin at maturity. The lignin play an important role at the maturity. So cellulose is insoluble in crystalline form, that is if they are arranged in an order fashion, or insoluble in amorphous form, that is it is a modified cellulose. If they are not arranged in a order fashion, that is amorphous forms. So what are the different cellulases? The enzymes which can degrade the cellulose are known as cellulases. What are the different cellulases produced by the pathogen to make its entry by breaking the cellulose? There are 4 different cellulose enzymes. Here you remember that you may get the chance of getting that question, that what are the different cellulases produced by the pathogenic organisms while making them their entry into the plant. First one is the cellulase 1, which attacks the native cellulose by cleaving cross linkages between the chains. So cellulase 1 attacks the native cellulose, and second cellulase also attacks the native cellulose only and breaks into shorter chains. Here cross linkages between the chains cellulase 1 is doing and cellulase 2 is doing, breaking into shorter chains. Then after shorter chains are formed, then CX enzyme plays an important role. These shorter chains are attacked by CX enzyme, then which degrade them into a disaccharide, that is cellobiose. Then after formation of cellobiose, then beta glucosidase comes, and this cellobiose is degraded by the enzyme beta glucosidase into glucose molecule. So glucose is a monomer, cellobiose is a disaccharide. So these are the four enzymes: cellulase 1, cellulase 2, CX enzyme and beta glucosidase. Then cellulase degrading enzymes play a very important role in softening and degradation of cell wall material and facilitate easy penetration and spread of the pathogen. Example we can see in higher fungi, that is Basidiomycetes fungi. This Basidiomycetes fungi examples and classifications already I covered in my previous lectures. So if you want those lectures, please go through them. And after cellulose, then we will learn about the hemicellulose, what it is and what are the different enzymes produced to degrade this hemicellulose. So hemicellulose are the major constituents of the primary cell wall and also seen in middle lamella and secondary cell wall. And this hemicellulose polymers include primarily xyloglucan, but also glucomanons, galactomanons and arabinogalactans. Don't confuse with these words, xyloglucan and glucomanons. These are nothing but cellulose backbone used to be there. They are linked with xylose, glucose, galactose and arabinose polysaccharides. This hemicellulose and hemicelluloses link the end of pectic polysaccharides and various points of cellulose microfibrils. In that microfibrils, in the previous picture I showed that hemicellulose is connecting these microfibrils. So it will connect the various points of cellulose microfibrils, and hemicellulases are the enzymes produced by the pathogens to degrade these hemicelluloses, and depending on the monomers released from this hemicellulose polymer on which they act, they are termed as xylanase. These are the enzymes. Xylanase, which will act on the xyloglucan, and galactanase, that which act on the galactomanons, and glucanase, which act on the glucomanons, arabinase and manase, and so on. Means these are the hemicellulases are different types on which substance they are acting. Example of the pathogen which produces hemicellulases are the Sclerotinia sclerotiarum and Sclerotinia fructigena. Just remember one or two examples for these pathogens which will produce these enzymes. Then after hemicellulose, we will learn about the lignin. Lignin is a very important part of the middle lamella because it will give strength to the cell wall. Then lignin is found in the middle lamella as well as in the secondary cell wall of xylem vessels and fibers that strengthen the plants. It is an amorphous, that is three-dimensional polymer which is made up of basic structural unit phenylpropanide. Phenylpropanide pathway, I think you knew about it. Anyhow, if you don't know, I will cover in the next lecture about this phenylpropanide pathway and what are the different units formed from this pathway. So how this lignin forms means, this is lignin forms by oxidative condensation between phenylpropanide units are substituted. Here you can see the cinnamyl alcohol. So that is here a picture for understanding purpose I gave pictures. Coumaryl alcohol, coniferyl alcohol and synapyl alcohol. Because of the oxidative condensation, the lignin will form from these things. Especially these ligninases, that is the enzymes released by pathogenic to degrade the lignin. Especially these are produced by white rot fungi which are belonging to the Basidiomycetes to utilize this lignin. Example, Xylaria, Chaetomium, Alternaria and Cephalosporium, they used to produce the ligninases. And coming to the cell wall proteins, because the proteins also used to be present in that microfibrils picture, I showed that some cell wall proteins are also present. So these cell wall proteins are similar to other proteins except they are rich in hydroxyproline. You have to remember this hydroxyproline, and we will see what are the different type of cell wall proteins like extensins, proline rich proteins, that is PRPs, and glycine rich proteins, GRPs, and solanaceous lectins and arabinogalactan proteins. These are the 5 different type of proteins we can find in the cell wall. These proteins are degraded by the enzyme proteases, and proteases are also known as proteinases or peptidases. And come after these proteins, then lipids and starch also used to be present in the cell wall, and various type of lipids we can find in the plant cells, either in the form of phospholipids or glycolipids. And these lipids contain fatty acids that may be saturated or unsaturated fatty acids, and lipolytic enzymes called lipases that which can degrade these lipids are known as lipases. These lipases may be either phospholipases or glycolipases, which hydrolyze lipids and release these fatty acids. These fatty acids will be utilized by the pathogens. And coming to the starch, starch is the main reserve polysaccharide found in plant cells, and we know that this starch is the very common reserve polysaccharide, and it is starch is nothing but a glucose polymer, and it mainly exists in two forms, that is amylose. Amylose is a linear molecule. In this picture you can see that amylose is a linear molecule, whereas amylopectin, this is a highly branched molecule, and this starch is degraded by the enzyme amylases. So because of release of these enzymes, the pathogen capable of degrade all these substances to make its entry into the cell, and it can utilize the food material from the host cell. So these are the different substances present in the cell wall. I hope you all understand about this lecture. If you have any doubts regarding this lecture, let us start a discussion. So you make your suggestions, or if you have any doubts in this lecture, just post in a comment section so that I will answer to all those comments. And if you want any specialized lectures regarding any topic also, just you make comment so that I will make another lecture on those recommended topics. And thank you for your keen interest in this video, and remember to like and subscribe for more videos. If you are finding this video is more useful to you, please share with your all friends. I wish you a great success, health, prosperity and happiness. Thank you, one and all.